This is the process of DNA replication. DNA is a type of nucleic acid that carries hereditary information that's passed on to generations of offspring. DNA is typically found in a double helix structure, where both matching strands are held in an anti-parallel orientation. The 5' end of the strand is matched with the 3' end of the other strand. DNA is made up of monomers known as nucleotides, and each nucleotide is composed of a nitrogenous base, phosphate groups, and deoxyribose. The sugars of each nucleotide are joined together with phosphodiester bonds. Each nucleotide in DNA contains one of 4 possible nitrogenous bases: adenine, guanine, cytosine, and thymine. These nitrogenous bases are separated into pyrimidines and purines. Cytosine and thymine are pyrimidines, while adenine and guanine are purines. Purines have a 2-ring structure, while pyrimidines only have a single ring structure. In DNA, adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds. Base pairing in DNA is highly specific, and according to Chargaff's rules, there should always be a 1-to-1 ratio of purine and pyrimidine bases. So the amount of adenine should equal the amount of thymine, and the amount of guanine should equal the amount of cytosine. The goal of DNA replication is to produce identical strands of DNA to be given to daughter cells during cell division. DNA is replicated in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. It occurs before mitosis and meiosis and during interphase, and more specifically during the S phase. DNA replication results in 2 identical DNA molecules from the one original DNA molecule. It is replicated in a semi-conservative fashion because both copies of DNA each contain one old original strand and one new strand. The main enzymes involved in DNA replication are helicase, topoisomerase, single-stranded binding proteins, primase, DNA polymerase III, DNA polymerase I, and ligase. Helicase uses free energy from ATP hydrolysis to break hydrogen bonds between bases, unzipping the DNA structure to prepare for replication. Single-stranded binding proteins bind to the parental DNA immediately after helicase unzips the strands, preventing the separated strands from rejoining. Topoisomerase relieves strain on the DNA molecule and prevents supercoiling so that helicase can unzip the strands in preparation for replication. Primase synthesizes a starter strand of RNA nucleotides, known as the RNA primer, for DNA polymerase to build upon. DNA polymerase III replicates DNA molecules to build new strands of DNA in the 5' to 3' direction. As DNA continues to unwind, one of the strands will not be able to build new strands as fast as the other strand. This is because the original strands are anti-parallel, and the DNA polymerase of one of the strands will always be building in the opposite direction of the DNA unwinding. The leading strand is known as the strand that continues to build the replicated strand in the same direction that the DNA is unwinding in. The lagging strand is known as the strand that builds the replicated strand in the opposite direction of DNA unwinding, since primers are continuously placed for the DNA polymerase to build new strands. The replicated fragments of DNA as a result of the lagging strand are known as Okazaki fragments. Next, DNA polymerase I replaces RNA primers with DNA nucleotides. Ligase connects the Okazaki fragments together by forming phosphodiester bonds. The ultimate result is 2 identical double helix DNA molecules.